I'd be grateful to have the opportunity of making this presentation here in 3rd Annual Doheny UCLA International Glaucoma Symposium. Since our laboratory is focusing on alpha imaging, today I want to present our recent findings about Bleb. I don't have financial disclosures. I received support from Japan Society for Promoting Science. So Bleb is the term for pocket of fluid formed between the sclera and conjunctiva after glaucoma surgery. The idea for glaucoma filtering surgery is to create a drain to allow aqueous fluid to pass from the inside of the eye to the bleb. Good bleb is to be an appropriate size, and bad blebs are huge or flat blebs. We always take good care of keeping the shape of bleb, but we know little about where fluid go after bleb. Bleb is something which is not new idea. It has long history as well as trabeculectomy. However, because of recent minimally invasive glaucoma surgery, Bleb attracts growing interest again these days. So this is the outline of the presentation. Firstly, I would like to talk about what's new in Bleb surgery. Secondly, what has been shown in the past. And thirdly, what we did to support prior research. And lastly, additional experiment we did about Bleb biology. These are the pictures of traditional Bleb surgery. Trabeculectomy and tube shunt operation have same concept, which leads aqueous fluid to subconjunctiva. And these are the new type of Bleb surgeries. Xen is approved by FDA and InFocus is under investigation now. Both use a small tube to create Bleb. Xen is ab interno and InFocus is ab externo. This is the result of Xen stent. This Kaplan-Meier graph shows the success rate of 3 years outcome. Success is defined as 20% IOP reduction. Left graph shows without medication. The right graph shows with medications. Both data shows that the success rate is similar to that of trabeculectomy. Additionally, Xen has the same problems as trabeculectomy. Xen also needs knee ring. Dr. Francis is going to show this in next talk. This is another reported IOP reduction of Xen. IOP is reduced around 25% in 18 months. These results show that Xen seem to work very well. Next, let's see what happened inside of the bleb. In this picture, tracer is introduced inside of the anterior chamber, and Xen is located here. This is bleb. This is the horizontal image, and this is the vertical image. You can see a lot of vessel-like structures inside of the conjunctiva. This was done in collaboration between our lab and Allergan in rabbits and Irvine. This is a picture of postoperative month 6 rabbit eye. There is then here, and this is the bleb. Fluorescent is injected into anterior chamber. So these lines are thought to be connected from bleb to blood vessel. And these lines seen here is thought to probably be a lymphatic vessel. So where does fluid go in a bleb? There are a lot of theories about where does fluid go in a bleb. Is it microzytal blood vessels, ovid, lymphatic? So what is shown in the past? You et al. Have shown that after creating a bleb, with trypan blue in conjunctiva, some pathways will come up in conjunctiva. And they have also shown that there are lymphatic vessels in conjunctiva using lymphatic marker. But they didn't show that this exact pathway is lymphatic vessel or not. So this is the picture of non-human primate, postoperative view 6. Xen is inserted here. So you can see the fluorescent from anterior chamber. This is Xen Bleb. And they also injected trypan blue from outside of the eye. This is trypan blue. So you can see some interactions between Xen Bleb and trypan blue Bleb. This means that the Xen shares outflow with whatever the trypan blue is using. This is the magnified image of connection between Xen Bleb and trypan blue Bleb. According to this image, trypan blue Bleb can be your substitute of investigating Xen Bleb. So what we did firstly was trying to show the exact vessel is lymphatic vessel or not. So these are the pictures of injecting fluorescent in conjunctiva, creating Bleb. Then linear-like structure. Yeah, linear structure showed up. So when we see this structure with anterior segment spectralis OCT, that structure has a cavity inside. When we see from vertical angle, there are bulbs inside the vessel. Fluid seems to flow to the right from the shape of the bulb. Also, the direction of the flow is consistent with this image. By the way, in arms and legs, our vein has bulbs, but above heart, there is no bulb. This is because bulb is for anti-gravity. If there are no bulbs in legs, it will be very hard to collect blood from tip of your legs. Therefore, it is thought that veins above heart don't need bulbs. So literally, you can say that it's lymphatic vessel if you see bulb inside of the eye vessel, but yet to be 100% proven, so we try to prove it. We started to do identification of the location. So where is the signal located? Is it conjunctiva or is it inside of the sclera? This is the picture after I peel the conjunctiva. There is strong intensity here because of this bleb. And here there's no fluorescent structure as we've seen before. This is the conjunctival piece after I dissected it from sclera. When we see this with excitation, the fluorescent structure in the conjunctiva, not in the sclera. Also, there is a lumen-like structure in the conjunctiva, as we saw before. So these are the pathology pictures. We use dextran as a tracer to do immunostaining. Dextran is made of sugar. It has an amide group, so it is trapped when fixed. Dextran has fluorescence, so we can see it. When we cut the section here, we see dextrin inside of the vessel. Then we stained it with PROX1, which is a lymphatic marker. So overlay image shows that this vessel is a lymphatic vessel. So we could finally say that this vessel is a lymphatic vessel. So then many questions come up to our mind. I'd like to show other experiments of Bleb works. The first question is how many vessels are shown in one Bleb? Is there a segmental lymphatic flow like segmental conventional outflow? And second question is how about in other species? So these are the videos creating Blebs. I'm injecting 2% fluorescent to create Bleb. You can see the fast flow here. It's interesting that sometimes it appeared and sometimes not. So how many lymphatic vessels will show up in one Bleb? Is it 1 or 2 or plenty of them? So these are the answers. This Bleb has 2 vessels, and this Bleb has also 2 vessels. This is a leakage, so it's not a vessel. So this has 1 vessel here. So this is a multiphoton microscopy picture. This side is the Bleb, so we can see more precise image of the vessel. And what is the distribution of lymphatic vessel in conjunctiva? Is it uniform or is it unevenly distributed like aqueous outflow? We counted these vessels with using spectralis OCT. It seems that nasal has more lymphatic compared to other 3 sections. I did t-test, but the number is low, so there is no statistical significance. But the number is still higher than other locations. So nasal Bleb can have more lymphatic vessel. If so, the nasal side will be more effective when we perform Bleb surgery. But there is no literature which compare nasal Bleb and temporal Bleb. But separately though, we know that lymphatic development starts on the nasal side of the eye. So nasal side can be more good location for creating Bleb. So we try this with cow and rabbit. Both showed like vessel-like structures as we saw it in pig. We will do this in human inoculated eyes in near future. This rabbit picture is given by courtesy of Susan Lee Halligan. So in conclusion, conjunctival lymphatics may be the main drain from blood. And our contribution is that we have proven the lymphatic aspects of earlier studies. Next question is, do Blebs require lymphatics? And if so, how to promote that? We are thinking of using VEGF-C and see if the lymphatic vessel will increase or not. So that's our new coming study. So thank you for your attention.